the lecture now. So how are you all?
Alhamdulillah.
Fine, sir.
I am
So how are
really
you?
I am really sorry for rescheduling your class because this morning I had a meeting with Bangladesh Academy of Science. They are offering a project, uh, commission project to me, and it is a very big project which we will work on food and nutritional security in the holder farmers in Borishal, Khulna, and uh, Rangpur. So in three divisions, there will be some agro-technological interventions, training of the farmers to increase their food security as well as nutritional security. Uh, this was a very important.
Congratulations, sir.
Thank you. This was a very important session because in last 30 years I have been tried to get funding from different donors, and I was fortunate to get lots of funding, but this time the donor came to me to offer the fund, so it was different phenomenon, and it was amazing. Anyway, we shall get a good funding for doing such activity at the farmers field to offer inputs and proven agro technologies to smallholder farmers in three uh, upazila, in three divisions, and each upazila we shall choose three villages, and each villages we shall choose a few hundred farmers as our target farmer, and offer them technologies for homestead, uh, as well as technologies for aquaculture and livestock, poultry, and crops. But anyway, it would be a bit different experience because I am basically a lab scientist. But what is going on in the field and how can we change the life of the farmers would be really challenging, and we shall have lots of opportunity to visit the farmers field, and so to see the reality of farmers life, and also how technologies are impacting the farmers productivity, income, food security, and nutrition. And Dhaka University also participate with this project to estimate the nutritional impact in the uh, immunity of the farmers and farm families. So they will analyze blood samples for vitamin D, zinc, and many other parameters. But anyway, it was a big negotiating process, and we shall have another phase of negotiation, that is the budget. But on principle, the project is going to be funded. So this is a good news today. And I had another good news to share with you. Uh, I collaborated with a professor at Kuwait to support a research of an MPhil student, and he worked in our laboratory for short time and generated some data on the biological activity of new nanomaterials they prepared by using silver and tin. Uh, Good news is that he's now in Australia working with a very big guy, my collaborator Yusuke Yamauchi, and they submitted a paper from uh, my students' MPhil thesis, and they got acceptance just on the day of my birthday. So he surprised me with a new birthday gift, that is acceptance of a journal research article to be published in Journal of Hazardous Materials, which has very high impact factor. 9 plus. So it was a very nice, good news to share with you. Anyway, I hope all of you are doing good. And in this group, uh, you are how many students? Four students, right? Four or five students? Five, five students. Okay, I shall offer you some of the opportunity during this course. If you are smart enough, you can grasp it. But let me start the second lecture and give you some more information regarding the ecological chemistry. Yesterday we discussed about semiochemicals as well as the pheromones. Pheromones are also semiochemicals. And semiochemicals, you already know that they are small molecules that affect the that have a powerful effect on the environment, and they are involved in crosstalks between and among different organisms living in an ecosystem. Pheromone is a semiochemical. Uh, There are lots of advantages of using semiochemicals in agriculture as well as in medicine and other industry. Semiochemicals, has adverse effects only on the target pest. For example, pheromone. Pheromone are actually not toxic substance, but pheromone trap you have seen yesterday. I have shown you pheromone trap we can use to lure or attract the different sets of an insect, uh, and then we can trap and kill them and control them. This is a safe way to selectively and specifically control an insect. And semiochemicals are relatively non-toxic and required in low amounts, incredibly low concentration. That means nanomolar or picomolar concentration, they are active. And this is why they are not toxic to the environment, not at all toxic to the other species. Even other species at such a low concentration cannot sense. Non-persistent and environmentally safe. That they are easily degrade in the environment. This is why they are safe for the environment. Difficult for insect to develop resistance against them because they are natural compound and signaling molecule, very specific, not creating any stress to the uh, organisms. This is why uh, against pheromone there is no report of resistance because they are the language of talking with each other. Monitoring of pest population with pheromones is often integrated in management program. Integrated pest management program pheromones are being used, and it is the safest compound. There are databases called, Pherobase. That means database of pheromone. The Pherobase is a freely accessible database of pheromones and semiochemicals, comprises several databases to provide comprehensive information about the pheromones and semiochemicals. Currently, the Pherobase contains pheromones and semiochemicals of more than 7,000 species and 3,500 semiochemical compounds. The Pherobase lists the occurrence of these semiochemicals within the various animal taxa, which is hard to observe in the raw literature data. So this is really a fantastic database, and information in this database are available like mass spectrometry, Covet's retention index, NMR, or synthetic procedure, chemical formula, 3D or 2D chemical structures of most of these compounds are included. So those who are working on pheromones or semiochemicals, this database is very, very useful. A Covet's retention index database for more than 8,000 organic compounds in 25,000 records with literature references are listed. a floral compound database of about 2000 floral compounds. Floral compounds means compounds derived from the flower. You know, flowers are diverse in color, structure, as well as in aroma. And all over the world, flowers are very, very attractive, for all human beings. And this is why flowers are used as a gift in the birthday or even in the funeral and all, human activity. 2000 floral compounds and their occurrences in 1,700 plant species is also listed. Another database of over 100,000 abstracts related to the Phero-based records is also included. You can browse the reference by journal, author, year. A database of the application of semiochemicals in pest management is also included and can be browsed by approach, region, country, and host. So this is the web link of this Phero-based database, and you can browse and find lots of exciting information. There are different classes of pheromones. First class is number one class of pheromones are sex pheromones. Sex pheromones are compound liberated by a female with a dual purpose of both attracting the male from a distance and also of inciting it to copulation when at close quarters. That means when it is close to the female, the male excited because the concentration of the pheromone are higher in the close proximity, and it triggers the expression of the different gene. So first one same pheromone do two purposes. One is attract the male, so attractant activities. Second one is inciting it to copulation. That is, different genes are involved in these two behavioral patterns. The same term also applies to substance produced by the males to excite the female. So pheromones are not only produced by the female, pheromones can be produced by the males also, but the functionally, fundamentally, they do similar functions. A second group of pheromones are trail pheromones. Trail pheromones, as the name implies, are used by the social insects to lay down an order trail which other members of the species can follow to guide them from the nest to a food source and back again. Trail pheromones are usually been used by insect. Insect, you can see if you carefully look at insect follow a common path, and if when they start to find the food source, and after coming getting the food source, they come back quickly in a shorter way, and release the trail pheromone so that other can follow the path and can get the food source, because insects are social insects. Similar social insects, there are others like honeybee and termite also. Alarm pheromones. Alarm is communicated to other members of the society by diffusion of the pheromonal vapors in the air. Most insect alarm pheromones are produced and delivered from the mandibular or anal glands or from the sting apparatus. Alarm pheromones, especially, honeybee or wasp or some bugs, they release such kind of uh, volatile compounds in the air to send the message to others that there is a danger. So alarm pheromone is also very, very important, and scientists discovered many alarm pheromones. Sex pheromones are probably the most widely studied group of insect allochemicals. Allochemicals means the chemicals that have effect on other organisms. Uh, they are the some sort of chemical language. Semiochemicals, allochemicals. Some plants produce allochemicals to stop germination of other plants. Allochemicals can be positive or negative. So insect allochemicals, pheromones are also allochemicals, and they have now been recognized and characterized in many different species. The simplest sex attractant is valeric acid. Valeric acid is a very small molecule, and it is produced by the female uh, insect wireworm of sugar beet, uh, and the scientists have discovered it. Majority of the pheromones are long-chain unsaturated alcohols, acetates, and carboxylic acids. So these are the features of pheromones: long-chain unsaturated alcohols, acetates, and carboxylic acids. Here you can see structures of some sex pheromones. For example, valeric acid. This is a very simple compound, and it is produced by the sugar beet wireworm, and it is a female as sex hormone. Similarly, wax moth Ephemia sociala produced armelain. This is another pheromone. Aphid produced Mayura bc nepeta-lactone. Nepeta-lactone is another compound. You see, the structures are very unique, and benzaldehyde produced by the moth, Leucaena impuris, 6-ethyl 2-methyl 3,4-dihydro 2 H pyran is produced by the Happy Ella's nectar swift moth. So these are some examples of pheromones discovered by the scientist, and you can regulate the insect if you want to control the population or attract the specific male or female insect. By using very small quantity of these compounds, you can do it, and most of them are they are basically volatile compound, and they are basically hydrophobic compound. Hydrophobic means, phobic means dislike, hydro means water. They dislike water. They are usually insoluble in water. So here, trail pheromones, let us know a little bit more. Trail pheromones are characteristically employed by ants, bees, termites, which produce them in a variety of special glandular tissues. Chemically, trail pheromones are of a variety of structures. For example, leaf-cutting ant produced a methyl 4 methyl pyrrole to carboxylate, and red ants produced a different structure. So you see leaf-cutting ant or red ant, they have a different trail pheromone in structure. Termites produced these, linear structure, unsaturated, hydrocarbon. So these are the examples of trail pheromones. Alarm pheromones. Production of alarm pheromones is often related to that of defense substance. Alarm pheromone is involved in defense of the insect against the enemies. In combat among social insects, the contents of the mandibular glands. Mandibular, perhaps, if you know the insect, organs. Mandibular means it is a part of mouth parts of the insect are discharged through the mandibles into onto the enemy, which is thus tagged as an aggressor. The sting apparatus. Some insect like wasp or honeybee, they have the sting apparatus at the back of the abdomen. There is a needle-like structure. So sting apparatus of bees and wasp contains several glands that produce alarm pheromones, and they contain lots of toxin also, sting apparatus. So alarm pheromones like formic acid, very a popular one. You can easily understand because obviously you experienced a bite of ant. When ant bite, you feel a serious itching and pain, and it is due to the formic acid. Similarly, Andecan, another compound also produced by the ant. Termite also produced some alarm pheromone, terpinolene. So they are structurally unique. Ant behavior. Natural behavior of ants have inspired scientists to mimic insect operational method to solve real life complex problem. And people studied a lot about the behavior of the ant because ant has some very unique behavior that are helpful to understand the mystery of their life and even related to our real life. By observing ant behavior, scientists have begun to understand their means of communication, yes, how they communicate with each other, ant-based behavioral patterns to address combinatorial problems, first proposed by Marco Dorigo, a big scientist. He studied a lot about the behavior of ant. Here you can see very interesting thing. Ants secrete pheromone while traveling. That means trail pheromone while traveling from the nest to the food, and vice versa, in order to communicate with one another to find the shortest path. You see, ant is living here. There is an ant hive, big, house for ant, and uh, ant, they move, search for the food, and they if they find the food sources here, what they do, they travel crisscross way, but when they find the food, they travel in the shortest distance, and others can follow. And so smart and really amazing. I can show you some more image. The more ant follow a trail, the more attractive that trail becomes for being followed. This is very interesting. Scientists did lots of experiment, by using a different pipe, tube, and put the food here, and to see whether and follow the longest path or the shortest path. And scientists found that ant always follow the shortest path. And this is really amazing. And their behavior of the ant. Here you can see the more ant follow a trail, the more attractive that trail becomes for being followed. So more ant when from the nest to the food source, you see people put an obstacle. Here is, the height of the obstacle is smaller than this height. So what they do, they can sense and they follow this path to get the food. You see, they put this type of barrier. So first time they follow both, and finally they decide this is the shortest path to get the food. So really smart, and lots of things we need to know how they decide. Basically, molecular mechanism is still unknown, but it is reasonable that to follow the shortest obstacle is the best for them. Uh, Route selection, ants are forced to decide whether they should go left or right, and the choice that is made is a random decision. Uh, Pheromone accumulation is faster on the shortest path, you see. The difference in pheromone content between the two paths over time makes the ant choose the shortest path. Uh, Positive feedback mechanism to arrive at the shortest route while foraging. So, a stigmergy or a stigmergetic model of communication is proposed for the ant. Different optimization problems have been explored using a simulation of this real ant behavior by using even computer. So these are lots of studies. If you browse the literature, you can find the ant behavior. So pheromones are species specific sensory input from the social environment that influence a definite behavior or a developmental process. So pheromone is really uh, species specific. And if you go through this paper, Carlson and Lucia in 1959 published a paper on pheromones, a new term for a class of biologically active substances published in Nature. So if you do not get access this paper, you can ask me, but by using, I think, scihub.se, you can easily get this paper. So pheromones we discussed a lot. Now we can discuss on another term, allomone. An allomone is any a chemical substance produced and released by an individual of one species that affects the behavior of the member of another species to the benefit of the originator but not the receiver. Very interesting. Alumon, uh, the producer gets the benefit but receiver not get the benefit, but it is a chemical substance and it is a target specific. Production of alumons is a common form of defense, particularly by plant species against insect herbivores. So different plants produce different, toxic substances or bad odor substances or repelling substances. They are called generally alumon. Many insects have developed ways to defend against these plant defenses in an evolutionary arm race, because when plant produce a toxin, uh, insect or herbivore can never stop themselves. They try to evolve to that the defense molecule, and there is an evolutionary arm race. One method of adapting to alumins is to develop a positive reaction to them. The alumine then becomes a kairomone. Very interesting. Kairomone means the organism like plant produces a toxic substances as a defense molecule to ward off the herbivore, for example an animal or other human or even an insect. But as herbivore has no way except eating that herb, so it starts to tolerate over the period of time. And finally what happened, that molecule is used as a marker to identify that particular plant, and it is no more toxic for the herbivore. Uh, at that time this molecule is called kairomone. Kairomone, originally it was a defense molecule. For example, bitter gourd, corolla. Corolla produce, cucurbitacin like toxic, bitter substances. But you see, human over the period of time, they adapted to that toxic, bitter molecules, and now bitter gourd, if it is not bitter, people would not take it. So bitter taste is unique for the bitter gourd, and it is now kairomone because people can tolerate that molecule very easily. So others alters the allomones to form pheromones or other hormones, and yet other adopt them into their own defensive strategies. For example, by regurgitating them when attacked by an insectivorous insect. Sometimes, a toxic molecules is produced by an organism, but for example, a plant, but insect sometimes, uptake that molecule, like butterfly, uh, uptake that molecule, or some insect, and they extract that molecule and deposit in their wing. Butterfly wings, you can see very warning color and beautiful color, and many insect has similar, beautiful color outside, and that color actually not just for beauty. That color is the warning predator birds, so that they cannot eat them, because that colors are the toxic substances they ingested from the when eating the plant. So really a fantastic mechanism. Plant produces defense molecules, and sometimes that defense molecule hijacked by the herbivore and use those molecules as their own defense from the predator. Predator means the bird and others who kill and eat the how to say, insects. So kairomone, this is a bit, messy slide, but you can go through what is kairomone. Already I told you, it is a semiochemical emitted by an organism which mediates interspecific interactions in a way that benefits an individual of another species. That means the producer is not getting the benefit, but another species can get the benefit from that one. So please go to this slide, because I will share it with you. So these are all about, some pheromone, but in the nature, uh, uh, chemotaxis, perhaps you heard about this term, chemotaxis. Chemotaxis, yesterday I told you, mosquito find us even in the darkest room by following the gaseous substances released from our body, and it is called chemotaxis. Chemotaxis means preferential movement of an organism toward a chemical source. And chemotaxis, in the evolutionary process, one organism can recognize another organism following the chemicals released from the other organisms. And chemotaxis is a very important phenomena in case of bacteria, in case of even fungi and many other organisms. One organism can recognize another following the chemical signal released from the one organism and perceived by other organism. In case of chemotaxis, one organism release a chemical signal, and another organism perceive that signal by the receptor, and it is established through the evolutionary process. In case of plant pathogen interaction, chemotaxis is very important. Even chemotaxis is present in the bacteria when they target the host specific organ of the host for colonization and others. So we shall discuss later on this chemotaxis in detail, but this is the image, actually a microscopic image I took when I was a master student in 1997 in Japan. Here you can see the root tip of sugar beet when you drop into the suspension of uh, a juice pool. Suspension of juice pool. Juice pool means some motile spore which can swim a very minute structure, 5 micrometer in size, you cannot see in the naked eye, and juice pores of a plant pathogen. So sugar beet is susceptible to a pathogen called Aphanomyces cochlearis. So Aphanomyces cochlearis release asexually millions and billions of juice spore, and that juice spore have the sensory transduction mechanism to find the root of sugar beet, because sugar beet release some chemical substances to the environment, and juice spore can sense, they have the receptor to perceive the signal. And when you put a root tip in the juice spore suspension, within few seconds you can see billions of juice spore aggregate on the root surface. And this phenomena is called chemotaxis. Sometimes they make this type of ball-like structure, huge number of juice spore insist, colonized in the aggregated on the certain position to increase the inoculum potential. And juice spore cannot actually infect the plant, but what they do, they change into cystospore, and then infection peg, they penetrate to the root tissue and infect the plant. So this was a very fascinating study when I was a master student at Hokkaido University in Sapporo, Japan, and the professor gave me this subject to discover the signaling molecule which attract the juice spore. And in the natural system, if we know the signaling molecules, then we can make an intervention to stop the damping of disease in sugar beet, because sugar beet is very famous for sugar production in the winter, how to say, temperate country. In our country, we get sugar from the sugar cane, but in most of the Western country, they get sugar from the sugar beet. And we discovered this compound. You see, very interesting molecule. We named it cochleophilin A. Cochleophilin A, because it attract the this molecule is very powerful at nanomolar concentration. It can attract the juice spore, and we used a particle. The particle is very small, 200 micrometer in size. People use it is a porous particle for gas chromatography. So when you add the extract of sugar beet on the particle and evaporate the solvent, and if you drop it into the juice spore suspension, you can see all the juice spore aggregated and then germinated all the germ tubes, which are far to the particle. They showed the germ tube tropism. So chemotaxis, differentiation, and the germ tube tropism, three jobs are done by these single molecules. And I had very interesting study. And this dummy, this particle can be used in the laboratory as a dummy of root. When you put the root, this is the root, and you can find the similar phenomena. So no need to use root, because in the rhizosphere means in the root zone of a plant, what is happening, you can do the research in the laboratory by using a simple method called particle bioassay method, to test any compound whether they are attractive to the juice spore or not. Even you can find the compound which are repulsive to the juice spore or toxic to the juice spore by using same particle bioassay method. And I published this paper in Plant and Soil, very good international journal. So this is all about today's lecture, and next time I shall discuss more about my journey on juice spore and interaction with its host, because Aphanomyces cochloides is not a fungus. It is a fungus-like organism belonging to the class of Oomycetes, but a terrible pathogen of spinach and sugar beet. Uh, and why it infects spinach and sugar beet but not other organisms, we studied its chemical ecology, and we discovered this compound, and we found that this compound are not produced by any other plants in this world except the host of Aphanomyces cochloides. This is a classical example of the co-evolutionary relationship between the pathogen and the host, how a single molecule, cochleophilin A, produced by the sugar beet, and which is used by the its pathogen as a signature or a marker to identify the host. That means we can easily call cochleophyllin A as a kairomone because producer is not getting any benefit. Rather, it is a problem for the producer because the pathogen, its enemy, is using it as a signature of locating the host and infecting the host. So this is all about today's information and chemical ecology. I guess you understood how fascinating, but the challenge is that to discover cochleophyllin A was not so easy. Dozens of scientists, over 10, 15 years, they worked to discover that molecule, but they failed to get it because that molecule present in the root of sugar beet at incredibly low concentration. As a result, most of the people they use few kg of root to extract the signaling molecule, but they couldn't purify by using column chromatography and other separation technique to get enough amount. Because to characterize a compound, you need 1 milligram or more amount. In our case, we discovered it first from the spinach root, and to get 1.2 milligram of that compound, we required more than 20 kilogram of roots. 20 kilogram of roots, spinach root is very small. You can imagine how many kgs of spinach plants we harvested to get such a small amount of compound. And after getting the compound in pure form, then the next job is to use spectroscopic method to elucidate the structure, what is the chemical structure, and it is a long procedure, not so easy. But interestingly, in that laboratory where I worked, when we discovered that compound, there were some synthetic chemists. They also prepared same molecules in the laboratory by synthetic chemistry by several steps of chemical reaction. And the synthetic molecule and natural molecule, we compared the spectroscopic data as well as biological activity, and we found identical activity, exactly same. That means that you can claim it is a kairomone and you really discovered the attractant. And it was a really a fascinating story. In chemical ecology, it is one of the brightest example. The chemotaxis signal, kairomone signal released from the sugar beet, how its pathogen use it as a signature or marker for locating the host, and how this molecule also trigger encystment and germination of the spore for initiating the infection. Really amazing an example. And based on our discovery, some scientists in Canada, they used this signaling molecule with the fungicide, pesticide. Juice pool attracted by the cochleophyllin A, and when reached to the source, they encountered serious toxin and killed by the toxin like that. So it is one kind of pheromone-like trap of the zoosporic organism, because in the soil pathogen, it is very, very difficult to control them. So this is all about from my side. Now you can ask a question or you can make comments on this, interesting subject of ecological chemistry.
Sir, I have a question.
Yes, Abdullah Rahat, please go on.
Sir, we know that in agriculture we use pheromone traps to deter insects, to insect control otherwise.
Yes, we use
And
a pheromone trap in agriculture, right?
my question is, can we use for other purposes these pheromones?
Pheromone trap for other purposes.
Sir,
Yes, if you want to
or
count
pheromones for other purposes.
pheromones for other purposes. Pheromones are specific bioactive compounds. In biology, people tested whether a sex pheromone of honeybee, whether it can any effect on other organisms or not. They are surprisingly very specific, and they didn't show any other activity. But if you want to do, calculate how many honeybees are present in uh, a region, because honeybee is very important for pollination and very important for agricultural productivity. So sensors of honeybee, you can calculate the use the honeybee pheromone or like that. But other purposes are not yet discovered. That we have no discovery so far a pheromone of an insect is useful for uh, you know uh, in other system or biology uh, where we can use it industrially. For example, some flavor compound produced by flowers, flower fragrance, they are excreted from the flower uh, to the air to attract the insect pollinator. But human, they love those fragrance, and a big industry, multi-billion-dollar industry is now perfume industry, and perfume industry is mostly based on those uh, flavor compound produced by the plants and other organisms. But in case of pheromone, we have no news of their use in other purposes. Who knows?
Thank you,
May
sir.
discover something other ways of using that pheromone. Yes, Rahat,
Thank you, sir.
welcome.
Sir, what about if, as it is a chemical compound, can we neutralize it by using another chemical compound? Just like I am being attracted by a bunch of honeybees, and I have a spray. I sprayed it, and it neutralizes the compound that used to communicate within the bees. So, can I save myself in this way?
Do you mean antidote? For detoxifying the uh, toxic substances from the insect or a snake, there are some antidote compound. But you mean if you have some compound that disliked by the honeybee, you can spray. That are called repellent. So repellent, you need lots of research to understand which compounds are disliked by the honeybee, or maybe some for toxic for their respiration, or for any other, or flying or something like that. Yes, you can use such compound, and if you can discover or prepare such compound, that would be good, and you can use them for handling the honeybee.
Thank you, sir.
No
But
worries.
sir, I wanted just to destabilize the natural compound, like pheromones, the
You
insect.
mean the destabilize?
Yes, sir.
Destabilize, what does it mean?
Like adding another chemical compound with that compound so that it changes its action, like
Yes,
with
it can be if you study well about the honeybee alarm pheromone, and if you have a highly active compound, then you can spray, and that compound maybe change into a, different structure, and loses its function of telling the alarm to others. Yes, it is possible. But in case of alarm pheromone, they work incredibly fast. Even you would not get time to change the molecule within few seconds, you will be the victim.
Yes, sir. Thank you, sir. I got
Welcome.
the point.
Any more question?
Yes, sir, definitely, sir. We know that in case of dairy farming, flies are the notorious agent which suck blood from the cattle. So in that cases, can we use pheromone technology to trap the flies? Or I want to mention that in our daily food, so when he goes to outside our campus, take food from the shops, so I have noticed that flies are several times sit within the foods and make contaminations. Contaminants. So what can we do in case of fly trapping? Have you
Yes,
understand?
Ruhul, I mean, it's really interesting question. Uh, Flies and sparrows, they are flies, sparrows, and crow. I found everywhere in this earth, even a temperate region like nearly Siberia, or even very hot and humid African countries. Everywhere you can, even in Europe, you can find the house fly or some flies, as well as sparrows and crows also. But anyway, flies are really very disturbing, as well as they transmit a disease when they move from a dirty place to the food. They sometimes contaminate, and they are really problematic because they can suck the animal blood or like that. So in that case, a pheromone trap, whether could be useful or not. Yes, pheromone trap obviously could be useful, but you need to discover a specific pheromone from the flies, but effective pheromone that attract the flies has not yet been commercialized, and it is a subject for research, and many scientists are working on it. That would be fantastic if we can discover such a pheromone to attract all the flies or specific flies in the especially livestock. uh, uh, then it would be a very good. Flies are really, cockroach. They are incredibly, adaptable to the changed environment, and they are powerful in the evolutionary process. Many animals or organisms, over the period of time and environmental changes, they extinct, but still flies, cockroach, crows, sparrows, they are everywhere in this world. Very good question.
Thank you, sir. So I have also noticed in industrial farming, commercial farm, they using thread-like structure above the cattle, and the flies those come around the cattle. They get attached with the thread. I think it may be pheromone or any other traps.
Yes, it is possible. I am not 100% sure whether it is available in the our super shop, but obviously you can search the literature and the pheromones of housefly or other flies. The, who are those are problematic for the livestock. Possibly scientists have discovered those pheromones, but I'm not sure. Some pheromones you cannot prepare in large scale due to the complex nature of their structure, and some are unusually fragile in nature. So, stability of compound is very important for commercialization. During transport, during packaging, during storing, they should be stable. But in case of pheromone, some of them are, very fragile. This is why commercially commercial production of those are a bit problematic. This is why people prepare synthetic analog of those compounds. which can mimic the similar function. Any more question? Fatima, Sumaya, and Shafi, any question?
No, sir.
Fatima,
No, sir, I have no question today, sir.
Sumaya?
No, sir, I have no question today.
Yes. Do you have question, Sumaya?
No, sir, I have no question today.
Okay. Then I should, offer you a big thank you because this evening we had a nice lecture on an interesting topic. I hope you enjoyed it. And what is most important, you know you are masters and PhD level students. It is hard for you to teach ecological chemistry because it is a very big and vast subject. So point by point, all the ecochemicals I cannot discuss in the classroom, but I shall go through the fundamentals and also try to inspire you to get into the subject and try to browse in the internet whatever you have question in your mind. Lots of literatures are available, and this is a fascinating subject of practical implications, as well as those who are nature lovers. You can understand how mysterious is the nature and how one organism is depend on other organisms and communicate with each other positively or negatively using very small molecules we call secondary metabolites. So thank you so much for attending the talk, and I shall obviously upload it in the YouTube, and you can get it again as many times as you need. Thank you so much.